Welcome to the second session of International Webinar Materials in Medicine, organized by Departments of Chemistry, Biochemistry, and IQAC of MES Asmabi College, P.Vemballur, Thrissur, Kerala, India. This is Mohammed Arij, your facilitator for the day. As you know, it is the time to convert the crisis of COVID-19 pandemic into oasis of opportunities by interacting with renowned experts of international stands. Today, we have Dr. V.M. Asma, Vice Principal and Head of the Research Department of Botany of our college, with us for giving few words at valedictory function of the webinar. She has been in the forefront of academic progress of this institution with more than 30 years of experience as a botanist and proven abilities of administrative skill. I welcome respectfully Dr. Asma to today's session. Dr. Sijo Chemannur, resource person for today, is scientist at Precision Nanosystem, Vancouver, BC, Canada. There is a lot to about his scientific career and field of work, which I purposefully delight to explain later. However, his wholehearted readiness to accept our request to give us an erudite talk has to be underlined. We were endowed to have you with us today for delivering talk. I proudly welcome Dr. Sijo to this online gathering. I happily welcome all the participants who have joined the webinar to remain with us till the end of the session, so as to facilitate your valuable feedback. Now, we are moving to the expert session. As I pointed out earlier, the resource person, Dr. Sijo Chemannur, is scientist at Precision Nanosystem, Vancouver, BC, Canada. He is noted researcher focused on novel role of GHPOK transcriptional regulator in oncogenesis. He is also a research associate at Fox Chase Cancer Center, Philadelphia, USA, postdoctoral research associate at University of Pittsburgh Medical Center, USA, and he has his PhD from National Center for Cell Science, Pune, India in 2014, and MSc in Molecular Biology from University of Pondicherry. His research areas are, as I told, include oncogene, nanoparticle-based drug delivery, gene editing, immunochemistry, and to list few. He has dozen international research publications with a very high impact factor. He has many awards of national and international standing, including NCS grant of Fox Chase Cancer Center, USA, NIH grant of Pittsburgh University, SRF of DBT India, Rajiv Gandhi Center for Award for PhD Scholar, CSIR JRF, PG Merit Scholarship of UGC, and first rank gold medalist for BSc in Biochemistry from University of Calicut. What I have said so far is just a briefing of the profile of Dr. Sijo Chemannur. We are lucky indeed to get him interact, and even that on very relevant topic, vaccine candidates for SARS-CoV-2. With all proud and respect, I invite Dr. Sijo Chemannur to deliver the talk. Over to you, sir.
Thank you for the beautiful introduction. I hope everybody can hear me properly.
Yes, sir. Yes.
So, Arij started out BSc from Krishna College, Guruvayur. So, I studied in Guruvayur College during my bachelor's. So I know Alice for a long time, and thank you for inviting me to this talk. Um, so, uh, in a broader perspective, I like to start from the very basics so that many people can understand and go towards little more complexity. And it's okay if you don't understand all of it, but try to understand some of the basics about the SARS-CoV-2 virus and what are the different new vaccine strategies. So without any ado, we can start our uh, presentation. So I'll try to present here. Yes, so this is the title of my talk, and it is the vaccine candidates for SARS-CoV-2. You can see the electron microscope picture of the SARS-CoV-2 in the middle. It's recently published in Lancet. And if anybody don't know what is SARS-CoV-2, it is the virus which causes the COVID-19 pandemic. So this is the virus which causes all the disease of COVID-19 all across the world. We are here in Vancouver, Canada. You are there many places of India and abroad, but not a single person in the civilized earth is not affected by this COVID pandemic. Everybody is affected. So at least we should have a general basic idea about what is SARS-CoV-2. So at least I hope this talk, the main objective of this talk is to address the question, what is this virus, what this virus can do, and some of the basic strategies, how people design the vectors, and so on. So before the talk, I would like to consider the fact that there's a huge amount of questions for COVID-19. I understand that there are a lot of questions. Everybody has questions. There is enormous amount of problems based on COVID-19, not only at the medical aspects, but also at the economic aspect, at the political aspects, and all across the world. So there are a lot of questions. Who made this virus? One of the major political questions now. And why this virus is so dangerous, if it is dangerous. And how this virus is affecting a lot of people, and what is this virus, and what I can do to prevent for not getting this disease, and if I get the disease, what should I do? Where should I hide? Because everywhere in the world is affected. So there's a lot of questions. They're bombarding questions about COVID-19. And there are a lot of monsters created because of these questions. But the biggest monster is the panic. If you see the comparison to the COVID-19, the panic is the bigger monster. People are so panicked at the economic aspects, at the political aspects, at the medical aspects of this virus, and a lot of panic spread around, which causes and affecting people's life. So there are different ways to overcome these obstacles. Humans find out different ways to cross these obstacles, and one major method is science. So I have some disclaimers, and the major disclaimer is what I'm talking is science or modern medicine. So I define science or modern medicine as well-documented growing body of knowledge, which humans can comprehend. In other words, I can say, as somebody said, magic is supernatural till we know how it works. And science or modern medicine cannot be magic or supernatural. So you cannot treat a patient with magic unless you directly know how it works. So this is how the modern medicine works. It cannot be magic. It cannot be supernatural. We should know how a drug works or how a virus works. So this is my disclaimer. And before going further, there are some basic understanding about biology is needed. Otherwise, it won't be I won't be having enough confidence to moving forward. So I hope most of the people know what is DNA. DNA is a genetic material, and it can be transcribed into RNA, ribonucleic acid, which is the intermediate readout of the genetic information. And from RNA, and these two are nucleic acids, and from RNA, we can get the functional protein through the process of translation. So I'm continuing with some more basics. So something which infects our body is called pathogen, and which includes viruses, bacteria, or many other mainly viruses and bacteria. And human body can have the ability to recognize or identify these pathogens as non-self. So there is self, that is the host, our body, and there is something called non-self, which is not present normally in our body, and that includes pathogen. So we have a tremendous capacity to identify something called non-self. And the immune response, the body defense, our host defense, are activated against this non-self pathogens, which can be a nucleic acid, a viral protein, or a bacterial protein. And once it is recognized, the next is a huge war. It's literally a war between pathogen on one side and host on the other side. In this case, human on one side and the viruses on the other side. So it's literally a battleground. There is military in our body, it is called immune system. How many of you can recognize this picture? It's from a movie called Matrix. I hope at least some of you have seen this movie. And it's named as Sentinel. So it has fantastic function. And the function is to recognize any other information which is they considered as non-self, and it passes the signals to the mainframe of the supercomputer, and it's named as Sentinel. It's exactly like that. Our body also has something called dendritic cells. It's ironic that it looks very similar, but it functions exactly the same. It identifies the non-self molecules and is reported to the main immune system. It's not only this, we have trained infantries, sharp snipers, sharpshooters, which blindly shoots at the pathogen or the non-self proteins or non-self viruses or bacteria. They are called cytotoxic T cells. They are blind. They just go and shoot the enemy. They don't think anything. They are very well trained military force. They just go and kill. They know nothing other than just go and kill the pathogen. And there are policing. There are police people inside our body. They have the ability to stop the pathogen. They always check that there is any foreign molecules or foreign pathogens inside the body. They are called as T helper cells. And there are tankers, that we have huge shooting tankers, which can be formed from differentiation from a B cell, which is called plasma cells. So they can shoot antibodies. That is our main arrows. So they shoot antibodies to viruses, and they can prevent or cause the death of any pathogen. So with this basis, I'm moving forward to the content of my talk. And the main content of my talk will be concentrated around the SARS-CoV-2, and that is the virus which caused the COVID-19 pandemic. And going forward, I will talk about the pathology of this virus, how this virus caused a disease in human. And third, I will talk about immunology of vaccination, or the question arises, many people ask why we have to vaccinate. What is the need of a vaccination? So I will address a little bit of that. And the main content of the topic is what are the different types of SARS-CoV-2 vaccines. There are many different types. We'll address some of the major types of SARS-CoV-2 vaccine which are in the making. What are the readouts of effective vaccination and strategies to be considered for vaccination? And what are the main forerunners of COVID vaccines now? So I hope to cover this systematically one by one. But if you have any questions moving forward, or if you're finding it a little more complicated while we go forward, you can stop or ask me questions. I'll be more than happy to answer those. So COVID-19 pandemic is caused by SARS-CoV-2. So there is a lot of resources available in the market, available in the internet. The best resources is from the NIH. 1 minute. So this is the, can you see?
No.
Yeah, so this is the website. You can search, anybody can search. It is covid19.who.int. They have all the updated resources about COVID-19. And if you can scroll down, you can see the confirmed cases, how many are affected. There's more than 1 million people already died because of COVID-19. There are 34 million confirmed cases. There is, as I told, there's more than 1 million people died because of this disease. And they have all the different sections of ethnicity and the countries affected and how many people are affected. American, Southeast Asia, Europe, Eastern Mediterranean, and the most affected countries. The first is United States of America, and second is the India, and then the Brazil, then the Russia. So if anybody has little more time and want to know the most up-to-date information, go back to this website. It's called covid19.who.int. That is the WHO website. It's very informative and it's available free for everyone. So it has all the countries' information. If you click India, it will give the information about India and so I'm not going into much detail. If you click India for the sake of our discussion, it gives all the information about how many cases we have, the situation of India, the deaths, how many deaths. So this is the most correct and most up-to-date information. And going back to the presentation. So it has, this website is a wonderful resource for anybody to look into and see by themselves. So the address is, again, I listed here, is covid19.who.int. So what is this virus? It's a severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2. It's very similar to SARS-CoV virus, which caused the SARS disease before from the Asian country. The one thing which you can notice or appreciate is the crown-like structure on its surface. And it resembles crown. That's why this virus is named as corona, which means it has this crown kind of structure on its surface. And it's very important for having this structure because it causes this infection. So it has a lot of different protein, which we go one by one in detail. So the first protein is the spike glycoprotein, which is also known as S protein. This is the most important candidate for vaccine. So it's very important to know if you're interested in any of the vaccination strategies for SARS-CoV-2. It's called spike glycoprotein S, and it is very important for viral entry. S1 subunit. It has two subunits, as I showed here. The red color is the S1 domain, which binds to the host receptor, and S2, this is the purple one, or the pink one, is the S2 domain, S2 subunit, which is necessary for fusion of viral or cellular membrane. So the next protein is N, which is the nucleoprotein N, which is important for replication and transcription. So the nucleoprotein is present inside a closed, it's not clear nucleus, but it is present near the genetic material. So that's why it's called as nucleoprotein. Next is the envelope or small membrane protein. I represent it as blue. It is necessary for assembly and morphogenesis of virion. It's also caused the pathogenesis. And then we have the genomic RNA, because it's a single-stranded RNA in this virus. It's a positive single-stranded RNA. Next is the membrane protein, which is or M, which is necessary for viral fusion, assembly, and the budding of propagation of viruses. So next topic, I would like your attention towards the evolution and spread of SARS-CoV-2. Many people have reported that it is mutated from bats, which is a silent host in bat, but it's mutated and it has an evolutionary advantage because of ecological stress, if not for the climate change caused by humans. There are a lot of research going on that aspect, on the ecological aspect, how these viruses are transitioning from animals to other silent host and then towards human. So there's a lot of research going on in this area. So in effect, this virus is mutated from bat, and it became some kind of evolutionary advantage. That means it has 3 mutations on its surface, or the crown, which I denoted as red. It has a modified spike protein now. So what happens now is it can infect and previously cannot infected cells, including ACE2 protein of human lung epithelium. So is angiotensin ACE2 protein, converting enzyme 2 protein, which is present in human epithelium. It can infect and go inside this lung epithelial cell or alveolar 2 type of cells, and then now it can infect human. So once it is infected by humans, it can be passed on through the liquid droplets, and this liquid droplets can go inside the human lung, and it can cause infection or inflammation, all those kind of diseases in the upper airway, as shown in this picture, and also in the lower airway or the lung, and which causes the liver fibrosis and death. And lately there are a lot of other symptoms. It can infect kidneys, it can infect heart, and it can also infect brain. There are a lot of reports coming up from US and other places that there are different strains are formed and it causes different disease symptoms. So what are the immune response to SARS-CoV-2? So once the humans get this virus through droplets, it can cause innate immune response. As I told before, it can cause the sensing of viral particles. It can cause activation of a lot of innate immune activators, which recognizes DNA, nucleic acids, or RNA. It causes secretion of type 1 interferons or inflammatory cytokines, dendritic cells priming, or active presentation. It may be a little complicated for people who don't know immunology, but that's okay. At least you should know that it happens by the activation of innate immune response, and then it causes the induction of adaptive immunity, which includes the cell types like T cells, B cells. As I told before, there are snipers, killers, there are police people, there are tankers. So it activates all this machinery inside our body. So it's just like preparing for war. The innate immune response are training the war people, and adaptive immunity is sending the war military into the battlefield. And the last part is immune memory to virus. So once the war is over, there are few people who will not die. They keep searching for the same enemy who is coming again and again. If they are coming again, these people, or the memory type of people, they stay for longer years to see if the enemy is coming back again. If they're coming back, they respond to them faster. So this can be the profile of the immune response. If the infection happens at day 0, the virus and antigen is shooted up in weeks. Accordingly, the specific T cells, or what I can say is the shooters, the main killers, are activated, which is shown as red in this picture. It go up in weeks and it comes down, but it is not going completely down. It stays longer for months and to the years. And the memory B cells, this is considered as the tankers. So they stays pretty longer time, for months to years. The memory B cells, the antibody stays longer, and memory B cells stays further, but in low amounts. So there are different phases of infection. The first, the virus can infect the ACE2 expressing nasal epithelial cells. So it first affects the upper airway, the mouth, salivary gland, the nostrils, and inside the ears and till the esophagus, upper respiratory tract. So this phase is asymptomatic or pre-symptomatic. Next phase is it infects ACE2 expressing cell type 2 in alveolar epithelial cells. So it is infecting the lower epithelial tract or the lungs, which causes pneumonitis disease syndrome. It causes pneumonia type of symptoms. It causes the activation of immune cells. And the last phase is the day 7 to day 10, which involves disruption of epithelial or endothelial barrier, complement deposition, and hyperinflammation. So in this phase, there's huge activation of immune response, immune cells, which causes, which includes neutrophils, cytokine storm, and whatnot, which is the most dangerous part for a COVID patient. So there are a lot of symptoms. This is from a CDC flyer. There are cough. This we all know now. The cough, so shortness of breath, difficulty breathing, fever or chills, or muscle or body aches, vomiting or diarrhea, new loss of taste or smell. And there are a lot more symptoms according to the amount of huge mutations which are happening. There are affecting the kidney function. It is directly affecting the heart. It is affecting the brain and many other cell types. There are papers came in Science which reports it directly affect the enterocytes in the intestine and blocking it and causing inflammation in the intestine. So, which is the cause, maybe the cause of the diarrhea. So there are ever increasing symptoms of this disease. And what are the treatment strategies available now? So we can look into that. The most important treatment is still the supportive care or respiratory support. That means 75% of hospitalized people of COVID-19, they require supplemental oxygen therapy. They need oxygen. Heated high flow nasal cannula oxygen if people cannot intake by themselves, or invasive medical ventilation. So we need a lot of hospitals for this. Targeting the virus and host response, there are different drugs available now. Antiviral, this name you might have heard a lot these days, is remdesivir. It is an antiviral antiviral drug which causes the inhibition of the replication, a very potent drug. And there are antibodies. That's why people are recommending to give plasma now, convalescence plasma. Anti-inflammatory agent dexamethasone, it showed very high promise in the clinical trials because it can cause the anti-inflammatory effect of cytokines, especially IL-6. Immunomodulatory therapies, many of the monoclonal antibodies, anticoagulants, antifibrotics or signal inhibitory molecules, which is mainly the tyrosine kinase inhibitors. So these are the many of the treatment drugs now available in the hospitals for treatment. And stop the spread of germs. That is the most precautionary measure we can do. Wash your hands often, wear a mask, cover your coughs and sneezes, and keep 6 feet apart. And mask work. Please wear mask. If not, you don't believe in the people, there is data available now. You can go to this Nature Medicine paper, and they have done extensive studies on wearing masks, not wearing masks, at different distances, very close, very far, all those kinds of studies. And according to the particle size, whether the particle size is more than 5 micrometers or less than 5 micrometers. In this study, you can see here on the far right that the particles, aerosol particles less than 5 micrometers without mask can spread very high viral load compared to the people who are wearing mask. So it's a clear data now available in the resources, open resources, that the wearing mask has a very potent effect as a preventive measure. So how vaccine works? So if you have a vaccine candidate, it should be first delivered inside the cell. That means it should go inside the cell, and it should do something to expose your vaccine protein or virus or DNA or RNA, which we'll explain in later. And this should produce proteins, various proteins, and which can activate the type 1 interferons or the innate immune response. And also, it has the ability to present this protein on the cell surface. So you can see this red proteins which are expressed on the cell surface, and these proteins can be recognized by the immune cells, which causes the cell-mediated or humoral immune response. So at least you understand this part, that the virus should be delivered inside the cell, and it should be presented properly to the immune response. So if we can meet these few critical criteria, then you can get an effective vaccine. So what are the different, so before going to the next step, I hope you understood till now. And if anybody has any questions till now, you can ask. Arij?
Yes. Okay. Anybody can ask questions. You can unmute your mic and raise your question. Yeah.
Hello. Hello.
I can't hear anything. Can you speak louder?
Sir, I want to know that since asymptomatic cases are increasing nowadays, so
I can't hear anything. Sorry.
so I have typed the question in the text chat box.
Sir, can you hear me?
Yes, now I can hear you.
Okay. Actually, I'm from MES College itself. This Dr. Nilima Shilev, she has typed a question. Actually, she was trying to ask the question to you earlier.
Okay.
Sir, okay, the question is, asymptomatic cases are increasing nowadays. So do they play a role in the spread of disease?
Yes, definitely. There are a lot of studies now work on younger people, especially the infants, toddlers, and asymptomatic healthy adults. So the rates are different of spreading, but the answer is yes. Any other question?
So there is another question from Firdausi Umar.
Yes.
He has asked, why is the virus spread so fast?
Why is the virus
spread so fast?
So it has a fantastic mutation, I would say, evolutionarily. This virus is it can recombine very faster. So it's called recombinogenic. So it can recombine and mutate faster, and it has this specific spike protein or the crown, the red crown I showed you before. So it can go and infect the cells very faster, very quickly. So that makes this more, I would say, evolutionary advantage I used before. So it can directly cause and infect human cells, which it couldn't before, but it started to have this fantastic function to infect the cells and the patients. Yes.
Sir,
Any other
Ramshia Ali
questions?
has asked. Yes, sir. Ramshia Ali
Yeah.
has asked why there is reinfection after recovery.
So yes, that is an interesting question. So there are few studies, I think, come from States and from Germany, and one from the East, Far East, Hong Kong, other places. The level of antibody response is not that high compared to the first infection. So normally what happens is, in a memory state, once the war is over, the infection is lower, the secondary immune response are very faster. But in this case, the priming or the initial infection is not activating the cells that high level to stop the second infection. But there are a lot more data has to come about how much severe that was, the secondary infection was in the same patient. Because most of the time, the secondary infections happens in asymptomatic patients. So people might have very mild fever, and there are a lot of studies are ongoing, especially with the medical frontline workers, where they have a continuous encounter with the patients. So still more data are coming out now, but the general answer is immune system is not coping enough to encounter this new antigen, new virus.
Sir, a lot of questions are actually popping up. Saujanya D, she has asked, nowadays the symptoms are so different. Based on the symptoms, vaccine varies or is it the same?
What is the question again?
Nowadays, symptoms are so different. Based on the symptoms,
Yes.
does the vaccine vary or is it the same vaccine?
So it depends on the vaccine strategy, I would say, because the lot of proteins on the surface are mutating. Yes, that's true. But we can, or we have the ability or technology to design vaccine in such a way that we can derive vaccine to a conserved sequence of the spike protein. And we can still efficiently block this virus infection. Yes, we can. There's still data coming. There is a fantastic paper came out 2 days back in September 29th about I will come into that, the Moderna vaccine later. But yes, we can. The answer is yes. Any other question? Or
Sir,
can we... Okay.
No, sir. There are a couple more questions. There is one
Okay.
more question for Dr. Nilima Silev again. Are the
Okay.
asthmatic people less prone to the disease since their cells don't express ACE2, which is a receptor for the virus?
I cannot say that, because the problem with the asthma patient is because already the type 2 immune response are very high. IL-13 level and IL-4 level are very high, and especially the type 1 response are less, especially the interferon gamma responses are less, which can be bad for this infection of coronavirus or SARS-CoV-2, because it's not only the rate of infection matters. Once it gets inside the immune system, how it grows and how it can be prevented. In the case of asthma, the type 2 responses are more the lung pathology is so disturbed. And this also depends on which grade the asthma is, whether it's a congenital disease or whether it is just a type 2 allergy disease. So the broad answer is, I don't think it can be protective for asthma patients.
Okay, the next question is from Pratiksha Tiwari.
Yes.
Can one virus affect more than one organ at the same time?
Yes, definitely. There's a lot of reports coming up from New York and other places that SARS-CoV-2 can infect multiple organs. There are few patients, young, healthy patients, who comes to ER or emergency room with stroke, just stroke, no other symptoms. And within 2 days, the kidney fails, heart fails, and then they die. So yes, the answer is definitely yes, it can infect multiple organs.
Okay, next question is from Monosha Priyadarshini.
Okay.
Cytotic T cells exist in our body, then why can't it act on SARS-CoV-2?
It's
Cytotic T cells exist in our body, then why can't it?
I understand that. It's
Okay.
so cytotoxic T cells. It need a proper activation to act. So it's just like trained soldiers. They are snipers. They can shoot and kill, but it need a command that, okay, you go and fight now. So that transition from command to the effective function, it need proper signaling. So in this case, we need a specific type 1 interferon gamma signaling for that activation. So we cannot generalize why there is cells and why it cannot kill, because it depends on the site of infection. For example, the upper respiratory tract is not mainly mediated by T cell immune response. It is mainly mediated by IgA response, which is secretory antibody response. In my context, it is the tankers which secrete some antibodies outside the cells, IgA. So it depends on the site of infection, how efficiently our immune response is reacting to it, and how efficiently the virus is. It depends on which side the war is winning.
Okay. Shirley Thomas has asked, even people with good immunity power is affected by this.
Yes.
Why is it so?
So healthy people are not immune to cold, right? We get cold. So getting infection doesn't mean that they are like weak. Getting infection is a part of a healthy immune response. The problem is whether our system can win the war against this virus. So if we need any more supplementation or hospitalized medication or other things to win over this war. So getting cold doesn't mean that your immune system is weak.
Sir, actually, there is a lot more questions coming up. Do you want to take a break or continue with your presentation?
So what I will say is I will take 2 more questions and then we'll move on. Otherwise, we won't finish.
Okay, sir. The question from Bonnie Thomas. Sir, is there any mutant strains of this virus? If yes, then what would be the challenges involved in vaccine production?
So there are a lot of mutations reported from different places of the world. There are some mutations from Italy, which is more, it caused more death than many other countries. It has a different strain. The strains in US there are a lot of people identifying more than 30 different strains now. So the answer is yes, there are mutations. And but it also depends on the vaccine strategy should also depend on the protein which we are targeting. So if the protein we are targeting is conserved, we can still win this war. So we have still hope to have an effective vaccine. It's over, Danya?
It's not over. Actually, Bhagyasri Paul has typed a very long question.
Wow.
Actually, I'm not able to understand the question. Can she unmute the mic and ask it? Bhagyasri Paul. Okay, I'll try to read it. I want to know, after vaccination, can any test is doing for the confirmation after 52 days of vacation, and any symptoms are showing after vaccination?
What is the question again?
Actually, I think Bhagyasri Paul, can you unmute and ask the question? I think she's trying to ask whether the vaccine work after 52 days. And is there any confirmation studies after
Yes,
52 days?
I will show some conservation, some studies for that vaccination. I will show in later slides. Don't worry. So I have a lot of questions. That means some people are understanding. So that's a good sign, right, Danya?
Yes, sir. Yes, sir. I think you can continue with the presentation.
Okay. Now we can go forward. I know you have a lot of questions without answering your questions. Bhagyasri is here.
Yes. I want to ask, after vaccination of 52 days, any confirmatory test is going, any confirmatory test after 52 days of vaccination?
There are a lot of confirmatory tests, and I will explain few of the very important confirmatory tests people do now. I will explain it in my later slides.
Okay. And one more thing I want to ask, after vaccination, which symptoms are occurs in a person?
Can you say that again?
After vaccination, some symptoms are occurs in a person, like fever, body acne, something.
Yes, definitely, yes.
Okay.
Yes.
Thank you.
Yes, it happens because there are different vaccines. I will explain. That is my next topic. I will explain what are the different types of vaccines people are trying out now. So if you have the patience, I can move forward and we can go together with that.
Okay. Carry on, sir.
Okay. Can I go forward, Danya?
Yes, sir.
Okay. So next topic is anyway. Thank you for your questions. I'm happy that you are, at least some people are understanding. The next topic is the type of SARS-CoV-2 vaccine, as Bhagyasri asked. There are types of SARS-CoV-2 vaccine in development. So there are different types of vaccines. Don't get confused by this. Don't get complicated by this. I'll explain it one by one. So there are inactivated vaccines, live attenuated vaccines, recombinant protein vaccines, replication inactive vectors, replication active vectors, all those things, blah, blah, blah. So we'll go one by one. The first is inactivated vaccine. So these are the SARS-CoV-2. You can see the presentation, right? Is it coming properly? Everybody can see the presentation?
Yes, sir.
Okay.
So, yes, sir.
Yeah, so the inactivated vaccine is the SARS-CoV-2 virus grown in cell culture, and then it is chemically inactivated. So that means the virus is produced. So virus is produced in the cells or the viro cells, and it is amplified in a cell culture facility, stored at -40°. And the second step is inactivate this virus. So how we do inactivation? Take the cell supernatant. The cell supernatant has this virus. So this virus we treat with beta propiolactone.
Sir, we cannot see the presentation.
Yeah, that's what I thought. So somebody can help me to put up the presentation, the coordinators. Arij? Hello?
We cannot see the presentation, sir.
You can see?
No, you cannot see.
Yeah.
Everybody can see the presentation? Or I can stop and represent.
Yes, we can see.
Presentation വരുന്നുണ്ടോ?
Wait, I'll try to do again. Now?
Yes, sir, it's visible.
It is okay.
It's okay now?
Yes, sir.
Okay, yeah. So these are inactivated vaccines are virus which are grown in cell culture. So we can make the virus in the cell culture and then chemically inactivate them. So viruses are grown in viro cells and we can amplify the virus. And then next step is inactivation of virus by some chemicals. In this case is beta-propiolactone. And we can validate this inactivation. And the third step is purification. So the inactivated supernatant can be constituted by buffer exchange column, then ion exchange column, size exclusion chromatography, and then we can then get the vaccine products. So there are a lot of candidates in this category. They are CoronaVac, or Sinovac, Biotech, Bharat Biotech. I think some excellent. Anyway, some Research Institute of Biosafety Problems in Kazakhstan. So the next is live attenuated vaccines. So this category of vaccines are pretty interesting. So what we do is we genetically change this virus. We have the SARS-CoV-2 virus, and we mutate many of these genes so that the virus cannot replicate that much inside a host or a human, but we express the spike protein or the S protein on the surface. So all the other protein I represented as dim because it is either lost or it is less expressed to prevent the replication of the virus or to prevent the cell growth or multiplication, but to express the protein which is necessary for the vaccination. So how this is formed? So this is formed by recoding the viral genome. You change the viral genome, edit the viral genome. There are a lot of different strategies to edit through CRISPR or many other different strategies. We can recode the viral genome, then synthesize this virus, infect the virus to the cells, and produce the virus. So there are different strategies we can use in a day-to-day lab. We use that in our lab for lentivirus and many other retrovirus and other things. So this is really possible. This technology is already here. And there are Codagenics and Serum Institute of India, Pune. They have a fantastic system of this vaccine. They are in developing this kind of vaccines. The next category of vaccines are recombinant protein vaccines. So what are these? These are just overexpress the protein, spike protein. If anybody working on the expression systems, you can express this protein in E. coli. For example, if it is more complicated, I won't go to much detail, but there are different vectors like PET systems, which can express the glycoprotein, recombinant S glycoprotein, and we can directly use this protein and inject into the patient and cause this vaccination. So there are many companies doing it, developing these kind of vaccines. Novavax, Sanofi, GSK is a pretty big company. They are doing this kind of vaccine for S protein baculovirus. And the next category is the crown or the recombinant RBD. So RBD is the receptor binding domain. That is the end portion of the crown which causes this infection. So those portion is very important for the infection. So we can express only the receptor binding domain which attacks the ACE2 protein. So there are many companies developing this kind of strategies with RBD dimers or RBD, that is receptor binding domain, expressing in baculovirus or expressed in SF9 cells. So other is the third type of recombinant protein vaccines are virus-like particles vaccine. So it is overexpressed, protein is overexpressed and it is present on the surface of a liposome or something similar to a membrane. So it's a membrane, lipid membrane with this expressed protein on its surface. So the SPY Biotech Serum Institute of India are developing this kind of VLP mediated vaccines. So these are under development. So the next kind of vaccines are replication inactive vectors. If it is getting more complicated, it's okay. Just this for your understanding. These are a different virus. Take a different virus. We have a lot of viruses which are commonly used in the laboratory settings, like adenovirus or adeno-associated virus, AAV, or Sendai virus, or retrovirus, or lentivirus. So take any other virus and express the S protein inside it. So it is any other virus which has the S gene inside it. That is what I'm trying to represent here. So red, I'm representing as a red for S gene inside this virus. So this S protein can express the spike protein. So there are many benefits for this because we don't have to handle the SARS-CoV-2 virus directly, so there is no handling issues. There is significant experience in production of larger quantities. We know how to make this virus. So we used to develop a lot of lentivirus, a lot of retrovirus, a lot of adeno-associated virus in our laboratory. So we know what are the facilities needed, and we don't need a very strict BSL-3 facilities, which is needed for the SARS-CoV-2 virus. So this method is fantastic in that aspect. It can stimulate effective adaptive immune response, but there is concerns for vector immunity. So the immunity can be developed against the new virus, if not for the vaccine. So that is the main disadvantage for this method. And there is huge amount of reports. This Lancet paper describing about the COVID vaccine from University of Oxford. So they have a similar vaccine with chimpanzee adenovirus. It's also known as CHADOX1 nCoV-19. It's mentioned here. So this is the University of Oxford vaccine. They published a paper in Lancet about the effectiveness of this kind of virus. So they have used the adenovirus to express the spike protein. So there are Cynobiologicals. They have robust vaccination strategies. Then the Janssen Pharmaceuticals, that is Johnson & Johnson Pharmaceutical associated companies, they have the adenovirus 26-CoV-S1 adenovirus for as a vaccine candidate. The next strategies is replication active vectors. So this is simple. So you just take any other vaccine, like for example, rabies vaccine or Sendai virus vaccine or Newcastle virus vaccine. You take that vaccine and put S gene inside that. Or take a virus which cannot cause any disease in human and put this S gene inside that. So we can do that. That technology is very much available now, and it can be delivered. So it can be delivered on the mucosal surface, so it can trigger the mucosal immunity, robust activation of immunity, and well-studied vectors like VSV, Newcastle disease virus. It is easily doable. There are a lot of papers regarding this. So this paper in Cell Host and Microbes, they studied extensively the effectiveness of SARS-CoV-2 under a VSV virus, and they can produce effective immune response. But they are not in clinical trials yet. People are waiting much more results. So there are vaccine candidates from Institute of Pasteur, Themis, and University of Pittsburgh and Merck. And the next candidates are viral vectors that display S on their surface. So these are the different vector like influenza virus vector, but we can make the S protein expressed on its surface. So what we do is we take the S protein gene, put inside some of its receptor promoter, and try to express the gene on the surface. So make it a corona protein on the surface of, let's say, influenza virus. So this strategy people are doing. It's more safer because only the S protein is on the surface. No other genome is there to cause this COVID-19. And antigen can be easily standardized, or influenza or rabies vectors can be easily used. They are in the preclinical stage now. So this is the strategy. That is, just take the influenza vaccine, put the spike protein on its genome, express the spike protein on the surface, like this side, express the spike protein on the surface, and then inactivate the virus using beta propiolactone, vaccinate the human patients, and then you get the antibody response. So it's pretty straightforward. So there are a lot of recombinant virus candidates from Bharat Biotech and as well as Mount Sinai and DV-based SARS-CoV-2 vaccine. So next strategy is DNA vaccine. This means just isolate the plasmid DNA from E. coli, just express the plasmid with the S gene and the mammalian promoter, and directly inject the plasmid into the human. So this is very much possible in large scale because we can make huge amount of plasmid DNA. It's very easy to make and it's more stable. But the main disadvantage is it is low immunogenic and it necessitates more complicated delivery systems. So this strategy is pretty straightforward. Take the DNA, inject into the body, and get the immune response. So there are many vaccine candidates. And other interesting recent development is the genetic information delivered as RNA instead of DNA, and then it is expressed in the body. So I said before about the central dogma, that is the first it is the DNA. DNA is coded into RNA and RNA into the protein. So what we do in this stage is just take the DNA, make the RNA in a cell-free in vitro system. That means make the RNA in a tube. We can make RNA in a tube. That technique is called in vitro transcription. And then encapsulate this RNA inside a lipid nanoparticle. So it is inside the lipid nanoparticle. So it is more stable. It's very stable. It can be used in many different conditions and temperatures without destabilizing it. It's a fantastic system. And many people are using this. It's cell-free, like manufacturing grade scale production of vaccines in very short time. Vaccine dosage requirement can be easily optimized. LNP, lipid nanoparticle formulation can be optimized for delivery systems because it's easily deliverable by lipid nanoparticles, long-term stability, and efficient activation of innate as well as adaptive immune response. So this is the paper which came in September 29 this year, 3-4 days back. This is the study from the group, the Moderna vaccine, mRNA 1273. So it is a mRNA vaccine. It has this lipid nanoparticles, and inside that there is an RNA. And they published this paper 29 September with effective immune response in older patients, like about 45 to 75 age group. So this 100 microgram dose is working fantastic, and they published recently in NEJM, New England Journal of Medicine. So these are the most important candidates: mRNA 1273, LNP, lipid nanoparticle encapsulated mRNA from Moderna, then 3 lipid nanoparticle mRNAs from BioNTech, Pfizer, from Germany. So these are the front runners of corona vaccines now. So these are the main diagram of COVID vaccines, different kinds of vaccines: DNA, RNA, live attenuated, inactivated, viral vectors, VLP. So there are many candidates for COVID-19 vaccines now under development. So how to test the efficacy of the vaccine? I think Bhagishree or somebody asked about this question. So we'll address few of them. There are many complicated tests and techniques which may not be the scope of this webinar, but I will explain few of it in a very quick time. I know time is running short. So this is ELISA, enzyme linked immunosorbent assay. Many people knows, if you know immunology, what is ELISA. So in effect, we have a petri dish, and we have a spike protein produced outside. So we can produce the spike protein outside using recombinant DNA technology or genetic engineering. We can synthesize this protein. So basically what we do is we'll put this protein inside the plate and then take the patient serum, dilute it, and patient serum has antibodies. So antibodies will go inside the plate and bind. So once that is bound, that bound antibodies can be recognized by HRP GOAT anti-human IgG. So this goes and bind on its surface, and once that is bound, it can glow. See? And once it can glow, this glowing can be detected. So anything can be glow. If it is glowing, we can read the readouts by spectrophotometer. So we can read by 570 nanometers reading. There is a lot of machines to read that, and we can read that. So this is one of the very basic techniques to detect the efficiency of vaccine. So you inject the vaccine, take the serum at different time points, like somebody asked about 56 days. People have asked beyond 100 days even. They take the serum, put that serum in different dilutions in the plate, and take the readout directly. There are more complicated techniques like this one. So this is SARS-CoV-2 nanoluciferase high throughput neutralization assays. It's a big name. Forget about the name. Basically, what it does is they develop a SARS-CoV-2 with a luciferase gene inside it. The lab has made it. It's available in many efficient labs. So they mix this virus with the dilution. So what happens is there is a competition between antibody and the virus. And now you infect this in a cell plated in a plate. So if there is less luciferase, that means the patient have more antibodies, so it has vaccination efficiency is more. If the luciferase is lesser, we can assume that there is more antibody in the serum. And if the luciferase is more, that means there is less competition and there is no antibodies in the serum. So there is ways to quantify the light emission by luciferase gene or the chemical which converted by this luciferase gene. So this luciferase can be, there are different techniques to quantify it, serum dilutions with 50% reduction in the RLU and all this. That is instrumentation part. The biologists don't have to break their head behind it. So there is focus reduction neutralization titer assay, FRNT assay. This again a GFP kind of color containing SARS-CoV-2 mixed with the different dilutions of antibody. Plate in as Vero cells, then layer it with methylcellulose, and then we can count the plaque formation or the colony formation of the cells, which can be quantified by microscope or by GFP containing a microscope, which can detect or ELISpot reader using FITC channel. So next is Industrialist cytokine staining. That means take the PBMC, put into a V bottom plate, and you take a pool of 50 more peptides of SARS-CoV-2, put inside this. Once it is put, PBMC can be activated in 6 hours into T cells. It can bind and detect the presence of cytokines, and it can be identified by flow cytometry. There's a technique called flow cytometry. We can take the readouts of the color based on the cytokine production of the T cells. So these are the different techniques to identify strategies to detect the efficiency of the vaccination. Next, what are the overview of vaccination production workflow? So the first step is collection of circulating viral strains. So we have to collect the viral strain or the swab or something. Genome sequence the genome, transfer this genome. So there can be sequence optimization and oligo design. We can synthesize the gene in vitro. We can assemble the gene, clone this. There are a lot of techniques available now to assemble and clone the gene. We can use traditional cloning. We can use gypsum cloning or golden gateway cloning to assemble the cloning strategies, manipulation of nucleic acids, large-scale vaccine synthesis, preclinical studies. We have to do this preclinical studies in mice, ferrets, or chimpanzees, and then the GMP or vaccination. So these are the main vaccine candidates now. The 3 main vaccine candidates. There are a few from China, but we don't have proper information, much information of that. But the more transparent, there are 3 main candidates. Moderna, which has from US, which has the candidate is mRNA, encapsulated in lipid nanoparticles. mRNA 1273 it is called. It is an LNP mRNA vaccine for full-length S protein or the spike protein with 2 stabilizing mutations. It is given in 3 doses, 25 micrograms, 100 microgram, and 250 microgram. The regimen is 4 weeks interval. And this vaccine is in the phase 3 trials in adults and older adults. The next is from UK, AstraZeneca company in collaboration with the Oxford University. The vaccine name is CH80 OXCOV19. Don't worry about the complication. It is CH for chimpanzee, AD for adenovirus, OXN for Oxford University, and for the COVID-19. So basically this vector expressing full-length wild-type S protein, and it is given as a single dose of 5 × 10 to the power of 5 PFUs or viral particles. And this vaccine are also in the phase 3 clinical trials. And many of the reports are coming up these days from the phase 3 trials. And the third candidate is from Germany, BioNTech Pfizer collaboration. This is again an mRNA-based lipid nanoparticle delivered vaccine. It has this receptor binding domain, trimeric version, so it has 3 receptor binding protein held by a T4 fold-on. It is given in 3 doses, 10, 30, and 100 microgram of RNA in 3-week intervals, and these candidate is also under the phase 3 clinical trials. So the other question I expect after this talk is why it is taking so long for a vaccine? Why it's taking so long? We are tired of this waiting, So it's because of this. It's a very strictly regulated process. So when the preclinical trials or toxicology studies are over in animal or chimpanzee or mice or ferrets, it is they have to the companies or the university have to submit the IND, that is investigational new drug compliances form to the FDI, and after that they have to do the clinical trials. The phase 1, which is done in a very small group of people, phase 2 expanded group of people, maybe in hundreds, and phase 3 is in 1,000 range of people. And there is process development required, and analytical qualification required, QC required from the companies, stability studies required from the companies. And after this, they have to submit BLA, that is BLA, or Biologic License Application. So after the clinical trials, they have to submit the BLA or biologics license application. From after BLA, they have to wait for the FDA approval, the FDA review. They will look into all the data and all the top scientists go in detail of all the information of the clinical data. And after that, they have to develop in a manufacturing stage. So most of the vaccines, or almost all of the vaccines now currently, or the most forerunners of COVID vaccines are in the phase 3 trials. So, and they are doing the phase 3 trials now, or they have done and validating the data, but it's not submitted for FDA approval. So at least we have to wait for, at the least, we have to wait for 3 more months for a vaccine candidate, and more time for its manufacturing grade development of vaccine. So that is my presentation. And I know you have a lot of questions, and I'm here to answer your questions if I can. Arij?
Sir,
Hello, I can hear you.
sir, yes. Actually, from the previous session, we have a couple questions more, but I think I have screened it and made it into 2. One is
Okay.
from Vrishali Goankar. She has asked, can I have some more details about immunity? How much time it requires for developing immunity in the case of COVID?
That is, if you ask about immunity, immunity is a very broad word. So any immunity happens when it first encounter the virus, but we don't know how efficient the immunity is. It depends on what level of immunity you want. You want no fever for you, or you want protection from the virus, or you want the activation of T cells or B cells, or you just don't want the disease in you. So if the question is, I just don't want COVID, how will I get immunity? It's tough question to answer, even for the experts. But I would say, as I mentioned in one of my slides about the phase of infection, so the 7 to 10 days after infection or is the most critical. That is the symptomatic phase. So if you pass that phase, your body develop the immune response to it, and the host wins. That is the answer I can give.
Thank you, sir. Another question from Pradishta Tiwari. I think she has actually posted 2 questions in between the session. So I think it will be better if she unmute and ask the question herself. Pradishta Tiwari.
Pratishta?
Actually, the question is, how did that translation process start? I think she was mentioning to the session. While the session was happening, she had typed it.
What is the question again?
How did that translation process start? That's why I asked her to. Pradishta, can you please unmute and ask the question? And the second question she asked is, sir, then in RNA, will it work as more of enzyme?
What is the question? Can you say that again?
Actually, Pradishta, can you unmute and ask the question yourself?
Danya, what is the second question? I didn't hear that.
Sir, then in RNA, will it work as more of enzyme?
Okay, so RNA can function as enzyme, but not all RNA can function as enzyme. So in the case of SARS-CoV-2, I don't think there is much data available now about the enzyme function of SARS-CoV-2 genomic RNA.
Okay. The next question is from Dr. Neelima Shivel. Is there any specific age group targeted for receiving the vaccine, or can it be developed to be administered in individual of any age group?
So I mentioned about the AstraZeneca study group and Moderna. They have the data available now from the older groups also. because the immune response is different in different groups. We might not get the same immune response from, we cannot expect the same immune response from the older group. So the dosage, there is a dosage difference in the older age group, and there is a tolerance level of uptaking vaccinations, side effects. For example, fever or shiverness or cold-like symptoms, or the vaccination site issues. So the tolerance in older group is different, obviously, compared to the younger group. So mainly it's working on the dosage level. Can we increase the dosage level so we can get a proper immune response from them? So that is the studies which are going on now.
Okay, thank you, sir. The next question is from Sarida Kancha. How do we know whether we are infected with more virulent or less virulent strain?
We can you say that question again?
She has asked, how do we know whether we are infected with the less or more virulent strain of the virus?
So it depends on few things, mainly based on symptoms. If you have more virulent strain, you have more severe symptoms. You have more further complications like multiple organ infections, and more immunization. Sometimes you may get less antibody titer, but more symptoms. So that is one suggestive of more virulent virus. And the readouts, as I mentioned, I discussed very few of the readouts. The ELISA can be used if we can activate immune system better. So the other problem is the virulence is a relative word. Virulence can be immunoevasive. So if a pathogen can trick a host more efficiently, it can be more virulent. So, and it can cause symptoms. So we have to be a little cautious about using that word.
Okay, thank you, sir. Then Shruti TP has asked, how many days does the virus stay in our body after recovering from COVID?
Yeah, that's an interesting question. There are reports coming up from the reinfected patients that the virulence stay in them for a long time, months, if not for more longer. So many of these patients which came in recent studies, they are asymptomatic. So they have very, very mild symptoms, maybe a mild fatigue, they're not feeling good or they're not energetic, but they have more virulence in them. They have more viral load in them. And especially in the younger infants and toddlers, they're completely asymptomatic, but they have the viral load. So those studies are still going on because we are still less than 1 year of this infection. We may need a lot more days to come to validate how long it can stay. But at least we know now that it can stay for months, if not more.
Sir, the next question is from Suma Soman. A lot of people are taking homeo medicines like arsenic album. Is it effective?
That is not within the periphery of this talk. That's a disclaimer I put because I'm talking about science and modern medicine. So as I mentioned before, the modern medicine go by data, and data means solid, well-documented evidence. The modern medicine say it's completely unethical to treat a patient without knowing what the medicine do. So in that context, within the scope of this talk, I cannot answer that question because I don't know how arsenic alba, that drug can work. Arsenic album can work because I tried to look into many of the data, but I couldn't get very precise, conclusive evidence for that. So I cannot answer that question. Sorry.
The next question is by Subhash Sarangi. History says that invention of vaccine for different epidemics in the past has consumed an average period of 10 years. How can we expect the vaccine so early and how effective would it be?
So
Is it clear?
as far as I understand, normally vaccine takes 10 years, and how you expect the vaccine to come faster now? That is a
Yes.
question.
Yes.
So there is something called accelerated FDA approval. So normally FDA requires more than 10 years to have a vaccine candidate, but now Food and Drug Administration has fast-paced it. So if you have a vaccine candidate and if you pass through the phase 3 clinical trials, there is a fast-tracked method now to get the FDA approval within few months. So most of the forerunners for vaccine candidates are finishing their phase 3 trials and applying for FDA review. So once FDA is reviewed, the process now is within months. It should be reviewed and finish the vaccine. So that phase is very much fast-tracked. So we can have an effective vaccine.
So there is a question from the same person. How effective is isolation or quarantine?
It's isolation and quarantine. Definitely, physical distancing is and the mask are the most efficient preventive measures now. There's no doubt quarantine has a very significant effect in reduction of the viral spread, but it depends on how efficiently you're strategizing your quarantine. Many times what people do is to pressurize quarantine not based on medical reasons, because of some political reasons or some other reasons. That is not the scope of this presentation, so I should not go into any of those topics. But yes, if you have any symptoms, please quarantine.
Okay, I'm screening some of the questions because we are having an overload, I think. Dr. Amit Kumar Tiwari has asked, can you shed some light on the toxicity profiles and therapeutic effects of the proposed vaccines?
Toxicity. So,
yes.
As I mentioned in some of my slide, I think here, yeah. So the first phase is the toxicology studies, and that studies are mostly done before the clinical trials. So all those things are in the preclinical phases, and they must do a lot of clinical trials, preclinical studies on toxicology. So that's why it's step phase. First preclinical, then phase 1 clinical to decide the dosage, then phase 2, phase 3. So before this, I have mentioned you about this. So if you can see, each vaccines have different doses. Like Moderna vaccine have 25, 100, and 250. 250 they may be putting for the older patients to get the proper immune response. But the dosage is different. Even though the Moderna and Pfizer they both have the mRNA vaccine, the dosages are different. Pfizer vaccine does not have micrograms of RNA. It has 10, 30, and 100 micrograms. So they have to standardize in each phase of the preclinical trials as well as each phase of the clinical trials. Did I answer the question?
I think yes. The next question is from Monosha Priyadarshini.
Okay.
She has asked, is there any chance of spreading of virus from a dead COVID patient?
That data is coming up. I'm not sure. There is very, very, very few data available now in the publication about that, but there is no conclusive evidence till now.
Okay, sir.
But there's a lot of, we can expect many of those kind of data, but I cannot give you an answer at this moment.
Okay. Pradishta Tiwari, I think she is not able to unmute. She has asked one more question. So my first question is, when you put the strain in body and it starts translation, so is there any specific place where it starts?
Specific place or
place
location?
where it starts. Maybe she means location.
Normally it first infects the upper respiratory tract, That means your nose, your esophagus, your pharynx, your ears, all those parts. And then when the days passes, it goes into the lower respiratory tract. And the question about translation, there is no specific checkpoint about translation. How the virus works is, once it is infected, the RNA goes inside the cell. It is transcribed by RNA-mediated RNA replication, and it is starting to bud up. So, the translation occurs readily. There is no checkpoint for translation inside the body, if not the infection rate.
Okay, so next question is, Prinsha Irshad has asked, what are the medicine recommended for the patients? As we know, vaccine is not produced.
I think I have a slide for that. I showed one slide. It's long back. Maybe I can find it. Then it will be much clearer rather than I'm saying a lot of things. It's in the early phase, right? Just bear with me. Yes, so these are the treatments which are available currently. So it has two different strategies. One is a supportive care, that is mostly the oxygen supply and invasive medical ventilation. But the drugs available are antiviral-based medicines, remdesivir, and antibody-based plasma, anti-inflammatory. Dexamethasone has a fantastic result now. There's a published paper on that. Immunomodulator therapies, anticoagulants, and signal inhibitory molecules. So these are the drugs available right now for COVID treatment.
Okay, so there is a question from Dr. G. Sharavanan. Cisplatin is used as a chemotherapeutic agent. Why cisplatin is not used widely? What is the limitation?
Cisplatin is a DNA damaging agent. It's a cancer drug, and I don't think it can be used effectively for this kind of treatments. Yeah, I don't think anybody has studied extensively about that, so I cannot answer that.
Okay, sir. I think that's the end of the question. I think a lot of comments, good comments are coming up in the chat box. I think
I hope I could answer some of the basic questions people might have in their mind. And if there are more questions, I each have my email ID, and they can ask and I can reply back.
May I ask?
Is it Saumitri?
Pradishta? Is it Pradishta? Yes. Saumitri Sahu. Okay, you can ask.
Madam, I am Saumitri Sahu.
Yeah.
Good morning, sir. Thank you for such an excellent talk. Sir, I have a simple question that the use of sanitizer, soap, or whatever which are available in the market, is it really helpful to prevent coronavirus, the use of these things?
So there are different strategies. Wearing mask is one, definitely. There are a lot of data available now that if you wear a mask, there is very less amount of viral spread. There is physical distancing data available. Sanitization of your hand, yes, it has an effect, but it alone cannot prevent the virus spread, right? It's a basic common sense that you use all the possible strategies to prevent this viral spread. If not for us, for our parents, if not for our grandparents, because they are the vulnerable group. We might get a cold, we might be sick for a day or two, or maybe a week, and we may be getting better. But for the people who are old, above 60s or above 70s, they might not be in a position to recover properly. So it's our social obligation for them to be protective and to take all the precautions that we can to prevent the spread of this virus, because it's affecting everyone. It's affecting you, it's affecting me, it's affecting everyone around us.
Sir, my question was that it is effective to inactivate that virus or anything?
So sanitizing your virus means you are preventing the spread of your virus through droplets into your hand. Into your hand means into your nose, into your mouth, into your own body. That is the idea. So if you sanitize and wash your hands, if there is any droplets which containing virus present in your hand, you can prevent it. You can stop it. That is the use of the hand sanitization. Did I answer the question?
Yes, sir. Okay, sir. Thank you, sir.
Hello?
Yes, sir. Thank you, sir. I got it.
Yeah. Thank you.
Okay. Almost your doubts are cleared now, I think. If there are no more questions, we have come to the closure of this session. Before winding up, I would like to extend my sincere thanks to Dr. Sijo for giving such an excellent lecture on this potentially relevant topic. Let's move on to valedictory session. I welcome Dr. V.M. Asma, Vice Principal of MES Asmabi College, for a valedictory talk.
Good morning, everybody. Respected, can you hear me?
Yes.
Can
yes,
you hear?
Yes,
Respected respected resource person of the day, Dr. Sijo Chemanur, scientist in Precision Nanosystem Canada, and HOD of Biochemistry Department, Sri Muhammad Ali GA, HOD of Chemistry Department, Dr. Ansar, IQAC Coordinator, Dr. Shafir, and all other participants of this webinar. Today, myself, Dr. Asma, Vice Principal of the college, representing our principal for giving a presidential address to the valedictory function of this 2 days webinar. This webinar is very much interested, and it is on the topic materials in medicine, and it is very informative in the current scenario of COVID-19 pandemic situation. Nowadays, what we can see is how it can spread and what are the measures we have to taken for preventing the spread of virus. This is because are getting an awareness through this type of about the pandemic situation. And the Department of Chemistry, Biochemistry, along with IQAC, selected the topic of relevance, and 2 resource persons selected who presented the topic. One is Dr. Payam Serintak, and today's resource person, Sijo Chemanur, is they need a special mentioning because they can able to present it very simply so as to all the participants got enough knowledge in this aspect. And it is actually a wonderful session because we know how to prevent this spread of the disease, and now we got a scientific idea about all these aspects. So once again, I congratulate the organizers for organizing this type of seminar, and all 2 resource persons and all participants for attending this webinar and made it a grand success. Thank you all once again. Jai Hind.
Thank you, Ms. Now it's the time to express my sincere thanks to all the supporters. I extend our sincere gratitude on behalf of organizers to our beloved principal, Dr. A. Bichu, and our Vice Principal, Dr. V.M. Asma, for their support and guidance. I thank Dr. Shafir P.S., IQAC Coordinator, Dr. Ansar E.B., Head of Chemistry, Dr. K. Keshwan, Head of Aquaculture Department, Dr. Denya P.R., and Prasoon T.P., Nodal Officer, BO Courses, for their wholehearted support. I would like to thank the audience for your patient listening and participation. Participants, please note the feedback form link is shared in the chat box. Please make sure your response. Each certificate will be sent to your email within 2 days. Thank you once again. Thank you all.